You may please start now. Thank you.
Okay, you ready for me to go then?
Yes, you can.
Okay, I will start. First of all, I think you recognize what's in the background. That's planet Earth. And the title, this was suggested to me, Research on Myxomycetes. Next slide. Well, there's a question that a lot of people have had. Certainly, I've gotten this question many, many times. What are Myxomycetes? If you went out on the street in New York City or Delhi or other places around the world and used the word Myxomycete, most people would not have the foggiest notion what you're talking about. Well, what are they? They're a group of fungus-like organisms. In fact, until about the 1960s or so, most people thought that they were fungi, thought of a special group of fungi, but the molecular biologists have corrected us and no way, they're not fungi, totally separate group. It is a small group. The total number of morphospecies, morphologically distinct species worldwide, is only about 1,000. It's actually a little more than 1,000, but more or less 1,000. To say that there's something that the average person knows nothing about, literally, very little, more apt to say nothing, that's certainly the case. I wouldn't want to quote any numbers, but I'd say out of 1,000 people, you might be lucky to find one person that knew what they were. Now, most of them are small. They're easily overlooked. Now, some of them are large, can be large enough to be seen easily, but most of them are not. One disadvantage they have, the common name which is applied to this group, slime mold. That's not an attractive common name at all. It tends to turn people off. Slime mold, who would want to study slime molds? Me, I studied slime molds, proud of it. They've been very good to me over my career. Next slide. This just simply depicts the fact that they are more closely related to, let's say, the common amoeba. You want to use that example, and not to the higher fungi like this Amanita. No, they're not related to the Amanita, except very distantly, of course. They're more closely related. In fact, the Myxomycetes truly are amoebozoans. That's a larger group to which they belong. Next slide. Why would anyone ever study them? Do you get major awards for studying them? No, not really. Well, first of all, there are places in the world where they've never been studied. I've traveled rather widely, and one of the main reasons I've done so, I wanted to go to places where Myxomycetes has not been studied before. We really don't know. In fact, we know now. Now we know more than we did 20 years ago, but we still have relatively incomplete knowledge about their patterns of distribution worldwide. For most groups of organisms, we can say with certainty that there are more of them in this part of the world, fewer in another part of the world. They're characteristic of a temperate zone, and so on. We didn't know very much about this. In fact, when I started my work, we knew almost nothing. Another thing, we don't know what they're doing out there in nature, at least not completely. The molecular biologists, using what is referred to as the molecular clock, indicate that the Myxomycetes, or the slime molds, there are more slime molds than Myxomycetes, but that's a group I'm focused on, they may be 1 billion years old. Now, something that's 1 billion years old and is common, Myxomycetes occur worldwide. They've got to be doing something. Now, some of what they're doing we now know, but a lot of stuff we do not know. The last point, how they interact with other organisms, is still largely a black box. We know that things are happening, but we don't know necessarily what's happening inside of that black box. Next slide. Occasionally, Myxomycetes get a little bit of publicity. This really astounded me. Obviously, this was a few years ago now, but in 2011, I got a call from a reporter at the New York Times, which is a major newspaper worldwide. He said, I want to do an article on slime molds. Couldn't believe it, but I talked with him. I provided information. I supplied the images that you see there, and they did a major full-page article on slime molds. It's amazing. In fact, when that appeared in the New York Times, I had former students that I hadn't heard from for decades get in touch with me. They saw the article. Wow, I guess reminded of the fact that maybe I was still alive. Okay, next slide. That's an ant. We have ants here in the United States, believe it or not. Well, there are ants worldwide. That ant is carrying a bit of a slime mold with it. Back to the nest. You say, wait a minute, can ants and other things feed upon slime molds? I'll use slime mold and Myxomycetes. I'll interchange, but I'm referring to the same group of critters. So could it be eating them? Yes, it could be. Next slide. That's a large fruiting of a Myxomycetes. It's a member of the genus Arceus, nice and red, colorful, and some Myxomycetes do produce fruiting bodies that are quite colorful. If you have very good vision, look in the middle of the screen, slightly to the right, and you'll see a beetle that's feeding on the fruiting body of that Myxomycetes. That's why it's there. Next slide. Ooh, that's a slug. Most people react in a very negative fashion to slugs. I guess I do to some extent. What is that slug doing? It's feeding upon the fruiting bodies of a Myxomycete. And if you look at the left, it's already fed there. It's kind of cleaned off all the spores out of those fruiting bodies. It's munching away. Next slide. That's what that particular Myxomycete looks like. It's actually a species of the genus Stemonitis, quite common. Kind of looks like hair in a sense, the same general morphology, but this one has not been found by anything that eats it. Well, I don't think it has. Sometimes when you see a fruiting like this, if you use especially if you have it available, which you probably wouldn't, some kind of probe or dissecting needle, and you sort of tease those sporangia apart, it's not unusual you find a beetle back in among the fruiting bodies feeding. They're rather secretive, but they're there. Myxomycetes represent a prime item in the diet, especially of some invertebrates. Next slide. Now, the Myxomycetes have a life cycle that I'm going to go over very briefly, but there are two phases or stages in the life cycle that the average person could see. The fruiting body stage or phase, large fruiting bodies, again, anyone would be able to detect them if they walked in the field. But there's also a vegetative stage called a plasmodium, which really acts like a giant amoeba. A really large plasmodium would look like this, but could be a lot larger. Can sometimes be more than 1 meter across. That's pretty good size. I've seen a few larger than that. I've had one person report that they saw one 8 meters across. I didn't see that, but they reported it, and I believe it. It certainly could be. Next slide. That's a little beetle, cute little beetle. Well, I guess. What is it doing? It's feeding upon the plasmodium. Yes, there are critters that feed upon the plasmodium in the same way that other critters feed upon the fruiting bodies. Well, there are actually some groups of beetles that feed on both. Next slide. This is a life cycle which I'm going to go over very briefly, A, B, C to whatever else. A, fruiting bodies. B, fruiting bodies produce spores. C, spores germinate to produce an amoeboid cell. That amoeboid cell can be flatulated or not. If sexual reproduction occurs, 2 of those cells fuse to form a zygote, G. That zygote develops into a plasmodium, which gets larger and larger and larger. There is a resistant stage that it can form, J, called a sclerotion. In fact, those amoeboid cells can form a resistant stage called a microcyst. That's E. Well, ultimately, that plasmodium transforms into fruiting bodies. I readily admit the first thing that attracted me to the Myxomycetes was a remarkable transformation from a plasmodium into a fruiting body, which can be an incredibly intricate structure. I wonder to myself, how on earth could this occur? You get something that's, well, icky, sort of gelatinous, and transforms to really what is a miniature structure, one of nature's most beautiful miniature structures, in fact. Next slide. This is what that amoeboid cell looks like if it's flagellated. These are unbelievably abundant in soil. They also occur in decaying wood. They occur in any place that you're likely to find the fruiting bodies of Myxomycetes, but you cannot detect the fact that they are there. You don't see them. They're very microscopic. Now you can culture them to isolate them, to find that they're there. And there are now molecular techniques, environmental PCR. There are ways of identifying that they're there, but you're not going to see them with the naked eye unless you have them in laboratory culture and you're looking at them with a microscope. Next slide. That's a large plasmodium transforming in nature. That's actually very common, large, with respect to the fruiting bodies, Myxomycete called Fuligoseptica. I know that in the English literature, it's sometimes called dog vomit slime mold, which is not, again, a very attractive name, but it's there. Kind of neat looking to me, but again, I study these guys. Next slide. One person looking at this or attending this presentation will recognize that and say, whoa, yeah, I know what that is. I fixed that up. That's a plasmodium being grown up in culture. And a plasmodium can get very, very large. And the person I'm talking about, who is listening to us, was involved in a project while she was visiting the University of Arkansas, working in my lab, seeing what she could do with a plasmodium. I mean, it's just, the plasmodium is just chock full of, as we say in America, full of lipids. And she was isolating those lipids. And one of the things we were speculating about, and she actually demonstrated, you can convert that into biodiesel, biofuel. Wow, that's kind of amazing. But it certainly has not been utilized to that extent, to any real extent, but it has some advantages over the other substances that we use to make biofuel. Next slide. That's a cute little guy. This is actually a Myxomycete called Himachal because it looked like a serpent, a snake. That's how it derived its name. This guy is just transforming from the plasmodium into the fruiting body. In fact, the main veins that occur in a plasmodium, I didn't point those out earlier, but there are thickened areas in a plasmodium, sort of a network, and the main veins, that's the term that we call them, actually the plasmodium material, the cytoplasm condenses on those veins, produce a fruiting body like this. It's a rather intricate, very interesting looking fruiting body. Next slide. Now, a typical Myxomycete fruiting body has a number of parts, just like a mushroom fruit, an agaric. If you're looking at a mushroom, it has parts that you recognize them, stipe and annulus and cap and so on. Well, for Myxomycetes, the parts aren't really quite the same. There's a hypothallus, which is actually a layer beneath the fruiting body that certainly represents sort of was the material left over when the fruiting body proper was formed. Excuse me. In a lot of Myxomycetes, they're elevated above the substrate and have a stipe, which can also be called a stalk. The actual mass, the spore mass, is called the sporotheca. The sporotheca is the mass, and whatever encloses it, in other words, it has a covering, which we call the peridium. Inside of the sporotheca, there are the spores, the spore mass itself. There can also be, in some Myxomycetes, some sterile elements inside of that sporotheca, and those sterile elements, which can form a network or be isolated, they're called a capillicium, referred to as a capillicium. Next slide. Wow, that must be a Myxomycete fruiting body. It is. A, remember the hypothallus beneath the fruiting body? B, the stalk or stipe? C, that's the sporotheca. D, that's a peridium, which is actually peeling off on this. You look at E, well, you can see the little masses of spores. And of course, in the background, well, actually, you can actually look at E and F. It can be a little confusing. I guess I would call F the spores, although spores are present in both instances. But that background, that network, that's the capillicium. So capillicium, the spores, the microscopic spores. Next slide. Fundamentally, there are four kinds or four types of fruiting bodies in Myxomycetes. Now, one type can grade into another, so they are not absolute. But most of the time, you can assign a fruiting body, if you run into it in nature, to one of these categories: sporangium, plasmodiocarp, ethalium, and pseudoethalium. So there are four types. Next slide. That's a Myxomycete fruiting body. Well, you can say, whoa, there are a number of fruiting bodies. You also notice something. This actually doesn't grade into it on a type or anything, but you can see that a number of fruiting bodies have actually fused together and share a single stipe or stalk. That's unusual, but it does occur in Myxomycetes. This is actually a Myxomycete called metatrichia. You don't have to worry about that, but you do get this fused configuration some of the times, and I certainly want you to notice what it is. Next slide. So, whoa, what's different? Well, that's a plasmodiocarp. Plasmodiocarps occur directly on the substrate, flat on the substrate, most of the time. There's one weird example it doesn't, but might as well forget it because it's so rare. This is actually Diderma effusum. Effusum means effused, flat on the substrate. This guy is quite common. It occurs on litter. And I will mention something. I'll develop this in a lot more detail later. Myxomycetes do exhibit some degree of substrate specificity. In other words, there are different kinds of substrates, and we're going to get into this, but a certain kind of substrate, you'll find certain species associated with it, but you may not find other species that you look for. But again, I'll come back to this. Next slide. So, whoa, this looks different. Well, this is Arceria cinerea. It's got to be one of the most common Myxomycetes in the entire world. And you look at this, you say, wait, I see those fruiting bodies. They look as though they're fused together. You see the things at the top that flare out. Those are each one of those is a sporotheca. But the stalks are joined, so they have a joint stalk. This species can occur this way some of the time, sometimes not. You find a single sporangium with a single sporotheca. And in older days, when taxonomy wasn't as well developed as it is now, people said, this is a different species. Well, it really isn't. It just takes two somewhat different growth forms. Next line. This is I call it a cutie pie. I find it a very attractive Myxomycete. It's called Hematrichia caliculata. And you look at it, the peridium, most of the peridium disappears, but it leaves a little cup at the top of the stipe. You can see that if you look closely. And that puffy mass of spores and capillicium, the yellow above it, looks like what we here in the States call cotton candy. This thing is only about 3 millimeters tall, and it's unbelievably common in the tropics, not so much in temperate regions of the world. So if you go to southern India, it's going to be all over the place. In a temperate zone in northern India, a lot less so. Next line. But in northern India, you're going to find this guy. This is Lycogla epidendrum. It has a distinction. This was the first Myxomycete described to science by Linnaeus. Now, Linnaeus wasn't a myxomycetologist. He didn't really study Myxomycetes. He thought it was a small puffball, and he described it in the genus Lycoperdon. But Linnaeus was wrong. You really shouldn't say that about Linnaeus, right? I mean, geez, yeah, putting Linnaeus down, we never should do that. But this thing, the fruiting bodies that you're looking at there, the largest one is about the size of your fingernail. It's a very common species of Myxomycete, occurs worldwide, except for the extreme high latitudes. Myxomycetes do not like really cold temperatures way up north and way down south. Next slide. Wow. If you're bright-eyed and bushy-tailed, that's a catchphrase of mine. People joke about that, but I've used it pretty much as long as I've been alive. You look at it and say, I think I've seen that before in this presentation. And somebody think, you know, I'll bet that's Hemitrichia serpula. Yes, it is. It's actually fruiting on a pond frond, actually in a semi-tropical forest. And that's a very large fruiting. You'd need both hands to cover what you see there. That's a very large fruiting, again, of a plasmodiocarp of Hemitrichia serpula. Next slide. Traditionally, and I'll say this at the outset, for a very long time, these were the 6 orders of Myxomycetes that were recognized by taxonomists. We now know that really the Myxomycetes do not quite fall out in this fashion. Some of those orders, and they're taxonomic orders, don't hold together. There are now new orders that are not on this list. But for most people, even those people who study Myxomycetes, I mean, there are few that study Myxomycete taxonomy. I mean, the people who are really serious about this, you could put them in a room together and there'd be a lot of empty space. Most of the people who work with Myxomycetes still use this traditional basic taxonomy. But the 6 orders, the Serratiomyxales, the Kinosteliales, the Lyciales, Trichiales, Stematales, Fissurales. You say, whoa, are they different? Yes, they're actually quite different. And if you're studying Myxomycetes, the first thing you learn, how to distinguish a member of one of these orders. If you assign something to an order, it's half identified. You've got a tremendous start. Next slide. Now, if you get a little bit into the molecular aspects of Myxomycetes, which I'm not really going to do this session, there are actually 2 major molecular clades, what we refer to as the dark spored clade or the light or bright spored clade. The Myxomycetes mostly fall in these 2 groups. Now, one of the orders doesn't. The Kinosteliales. They seem to represent a basal group that split off before these 2 major clades evolved. But then they come to the Serratiomyxales, they're not really Myxomycetes at all. We now know that. It's a sister group of the Myxomycetes, but they still have a lot of characteristics in common with the Myxomycetes, and the people who study Myxomycetes still place them with the Myxomycetes. When I go out collecting, I still collect members of this Serratiomyxales like anything else. Next slide. That's Serratiomyxa fruticulosa. Exceedingly common all over the world, except at the high latitudes. But anywhere else, for example, in northern when I was at Himachal University, actually waiting for it to rain when I first got there. The monsoons hadn't arrived. It was dry. I couldn't find any Myxomycetes, or found very few. When the rains arrived, Serratiomyxa fruticulosa was all over. Next slide. Some basic definitions. This is an informational seminar. I'm showing some nice slides, or what I think are nice slides, but I want to convey some information too. There's some definitions, and I came to myxomycetology as an ecologist. My training was in forest ecology, and then I started studying mycology, and as part of that mycology, my professor introduced me to Myxomycetes, And then literally I left my professor behind and became a myxomycetologist. All right, habitat, a particular type of ecological community. You say, what's that over there? That's a forest. Oh, no, that's a grassland. That's a desert. Habitats are easily recognized. No big deal on that. Microhabitat. What is a microhabitat? A microhabitat is one component of a larger habitat. I'll use the forest as the best example, or the easiest to understand example. You walk into a forest. You say, well, there's a tree. Is there bark on the tree? Yes, there's bark on the tree. That bark on the tree is a microhabitat. You say, wait a minute, I'm walking across dead leaves on the forest floor. Ooh, that litter later is another microhabitat. Ooh, that tree has died and fallen to the ground. What's that? That's a decaying log. That's another microhabitat. How about these twigs I see on the ground? That's another microhabitat. What about these dead plants? These dead plant parts that are still attached. They're dead leaves, for example. They're still hanging on. That's aerial litter as opposed to ground litter, the litter that you're walking across. And I'll get to this. There are more microhabitats than what I have used as examples here, but I think you get the concept of what I'm talking about. Next slide. Are there lots of microhabitats other than what I've mentioned? Well, I think there are. Coarse woody debris is a more technical term for decaying wood. Ground litter, that's a sort of an inclusive term. The stuff that on the forest floor, well, it doesn't have to be a forest. Ground litter can occur in a grassland or a desert, lots of other places too, but it's on the surface of the soil of the ground. Bark surface of living trees, I've talked to you about that. The dung of herbivores. What's dung? You see a cow eating grass. That digestive system of the cow processes that plant material. What dung is nothing more than processed plant material. So it's fundamentally plants. That plant material decomposes. Bacteria are involved in that decomposition process, and that gets us back to the fact that Myxomycetes feed primarily upon bacteria. So plant material, bacteria, Myxomycetes, yeah. There are Myxomycetes that are known only from dung. Aerial litter, lianas or vines, the plants that actually have their foliage, as it were, and up in the canopy, but stems that extend all the way to the ground. Not very common in temperate forests, but abundant in tropical forests. Well, lianas actually have bark, and what you find on liana bark tends to be very different than what you find on tree bark, believe it or not. Inflorescences of large herbaceous plants. In the tropics, there are plants that produce very large flowers or groups of flowers. An inflorescence is nothing more than a group of flowers. Very large, flashy inflorescences. Believe it or not, there are Myxomycetes that occur in those. And some of those Myxomycetes, in my experience, I've never found on any other substrate in a tropical forest. Don't know what's going on. That's a little bit of a black box. We think birds disperse the spores of those Myxomycetes that occur in inflorescence. We don't know. It's not been documented. Next slide. Ooh, epiphytes. In tropical forests, where the plants are evergreen, they stay on the tree long enough, and of course the conditions are quite moist and hospitable for plants growing. Liverworts, especially liverworts, in other words, bryophytes and lichens, develop on the surface of the leaf, the upper surface of the leaf. Those are called epiphytes. A leaf is a tiny community. You say, whoa, if you take that single leaf back to the lab and put it in a culture dish and keep it moist, some of the times you'll see that there are actually Myxomycetes that were existing in that tiny micro ecosystem. Wow. Bryophytes, where bryophytes are abundant, mats of bryophytes that occur on the forest floor or mats that accumulate on the branches of trees. There are actually Myxomycetes that occur there. Again, so Myxomycetes that tend to be associated with that substrate. Soil, dirt to the layperson. Myxomycetes are abundant in the soil, and we think they are major factors in nutrient cycling. That's something that's come out in the last couple of decades. Without the Myxomycetes, nutrient cycling would not be nearly as efficient. They play a vital role. Again, not appreciated. Most textbooks do not even mention this fact. I'm talking about ecology textbooks. They just haven't caught up with the ecologists yet. Submerged plant debris. You go down to a pond or a stream, and you see stuff that is leaves, twigs that have fallen in the water and been underwater for a while. Believe it or not, you can take those back to the lab and culture them, and you'll get Myxomycetes. There are some Myxomycetes that are even known to fruit underwater. It's amazing. Canopy soil, whatever canopy soil is. Well, in the tropics, on larger branches of trees in a tropical forest, I'm talking about wet tropical forest, you usually get a thick mat of bryophytes and vascular epiphytes forming, and they're there for a long time, decades. Beneath that mat of epiphytes, they develop something we call canopy soil. It's really very much like soil. If you look at that and you take samples of it and take it back to the lab, you'll actually discover that there are Myxomycetes there. They live in that microhabitat. Not been studied very much, but studied a little bit. Next slide. Now, if you've been paying attention, I'm sure all of you have. You're like my students. No, you're not like my students. My students wouldn't necessarily be paying attention. So those look like dead leaves. You're right. That's a litter layer on the forest soil. This is actually an image of dead leaves on the forest floor of a tropical forest in Costa Rica in Central America. But a forest, pretty much anywhere in the world, the leaves would look something like this. You'd recognize them as leaves, and you'd recognize that as a litter layer. Next slide. In the same way, most people would say, that's tree bark of a tree. And a lot of you say, look at all the lichens on it. Well, if you look closely, there are a lot of bryophytes on it too. They're just not as obvious. But that bark is an incredibly good microhabitat for Myxomycetes. There are at least 100 species of Myxomycetes that are known to occur in the bark microhabitat. Now, what do? You say, well, do you collect them in the field? Not normally. What you do is you collect samples of bark and you take it back to the laboratory, and I'll tell you a little bit more about that in a moment. Next slide. Remember I mentioned the term aerial litter? That's an epiphyte, a plant that grows upon another plant in the tropics. And you look at the, oh, those leaves, you can see the arrows pointing out, those look very dead. They are very dead. They've been dead for a while. What you do, you collect those, take them back to the lab, and you'll isolate Myxomycetes. There are Myxomycetes associated with those. In fact, in tropical forests, oftentimes, there are more Myxomycetes associated with aerial litter than there are with ground litter. Think about that. More up top than on the ground. We think there's a pretty simple explanation for that. In a wet tropical forest, the ground litter stays wet most of the time. And Myxomycetes have to breathe in the same way that we have to breathe. I tell my students, I use an example, which is really absurd, but I said, well, if I put you in a swimming pool and I kept your head underwater for 20 minutes, what would happen? And they know the answer. They'd be dead. Well, Myxomycetes, literally in a swimming pool, which is the ground litter would represent because of the film of water, they would die. These guys, as soon as the sun comes out, aerial litter dries off. It's a wonderful habitat again. Next slide. Those are lianas, vines, as I've already indicated. They're very good for Myxomycetes. Now, there are some vines in the temperate zone, and I've had some of my students look at those. They're not nearly as productive as the lianas in the tropics. Not nearly as good, but there are Myxomycetes there. Next slide. That's an inflorescence of a heliconia. It's a neotropical group of plants. They're planted as ornamentals elsewhere in the world, so I suspect some of you have seen them. Now you look closely, you say, what's that white stuff? You look very close, you say, those look like fruiting bodies. They are fruiting bodies, the fruiting bodies of a Myxomycete. That's Pistrum didermioides. That's an example of one. I've done a lot of work in the neotropics. Tropics elsewhere in the world too, but a lot more in the neotropics. This thing is common on the inflorescences of Heliconia. I have yet to collect it elsewhere in the neotropics, which is kind of amazing. Next slide. I can't remember what this is. Yeah, I can. That's dung. Now, if you want to collect Myxomycetes on dung, wait until the dung is dry and a gray, crusty surface forms. But you take that back to the lab, and sometimes if you collected 10 samples from that and took it back to the lab and cultured it, my guess is that a lot of time you get 10 samples, say 10 petri dishes, 10 samples. 9 out of 10 or 10 out of 10 would yield Myxomycetes. Myxomycetes are very commonly associated with dung. Next slide. How do you study these guys? Well, there are two basic ways of studying Myxomycetes. Sometimes one is more appropriate than the other. During the winter here where I live in Virginia, I couldn't very well study Myxomycetes except using that first method or the second method. You can go out in the field and collect fruiting bodies that developed under natural conditions. If there's been a period of rainy weather, wait a couple days, go out, and during the growing season, during the summer and early fall, you're more likely going to find them. But if you can't do that, you go out and snow can be on the ground. Go out, collect bark, for example. Take the samples back to the lab, put them in a petri dish or some other suitable container, moisten them, and put them aside, and you can get Myxomycetes, literally collect Myxomycetes that way too. Next slide. Petri dish, usually we put a disc of filter paper in it. There'd be other things too. Paper towel works well too. Those are pieces of bark. They have yet to be moistened. You actually moisten the bark. Fill that petri dish with maybe about 2 millimeters of water, 2 or 3 millimeters of water. Leave that overnight. The bark soaks up a lot of it. Next morning, if there's excess water, pour that excess water off, put it aside, start checking it with a stereoscope. About 10 days, sometimes a week, 10 days later, you're going to have Myxomycetes start appearing on that. Sometimes, and this is not unusual, I have prepared bark cultures which have yielded 5, 6, or 7 different species of Myxomycetes in one, what we call moist chamber culture, which is what you're looking at there. Next slide. Normally, if you're going to do sampling survey work, you prepare a lot of moist chamber cultures. I tell people something they do not believe, but one time when I was younger and had a lot more energy, I guess, I prepared 700 moist chamber cultures at one time. I literally ate, slept, and checked moist chamber cultures. I had no time for anything else. I would never do that again. I mean, got a lot of data, but no, you don't approach it that way. Today, since I'm retired, if I do 30 at one time, that's about as many as I go. Maybe 50, but no more than that. Next slide. You say, what's that? Well, that's a Myxomycete. In fact, it's a kind of Stelium minutum, incredibly abundant. If you prepare moist bark and moist chamber cultures, at least in the temperate zone, you're going to get this. It's not as common in the tropics. It actually does occur in the high latitudes, not the ultra high latitudes, but it gets pretty far north. And you look at it, it has a nice little stalk. You're looking at, has a spore mass at the top. Minutum means small. When this thing was described, it was thought to be the smallest of all Myxomycetes. The kind of Stelium minutum. Well, it's not. There are quite a few species that have been described since that are smaller, but it's still a kind of Stelium minutum. Next slide. You couldn't see this unless you had a microscope, but this is mounted on a microscope. You can actually pick out the individual spores. You say, well, wow, I could count the spores if you could spread them out, and you could indeed do that. For a large fruiting of a Myxomycete, you're talking about tens of thousands of spores. No way you could count the number of spores. You also see that at the base of the stalk, to collect this, a little bit of the substrate material was picked off. In fact, that's the easiest way to collect this little guy and put it on the microscope slide. But that gives you an impression of what you're looking at. And this is not a large Myxomycete, but you can just barely detect it with the naked eye, but on a stereoscope, it leaps out at you. I mean, it's plenty large to look at in detail under a stereoscope or dissecting scope, whichever term you're going to use. Next slide. Major terrestrial ecosystems. A lot of very different places. And one of my goals in my research life, I was interested in doing this for non-research reasons too. I really wanted to study Myxomycetes in every major type of terrestrial ecosystem. And I did it. Took a few years. High latitude polar regions. Yep, there are Myxos up there. Tundra. Well, you go far enough north, you're going to run into tundra, polar tundra, but there's alpine tundra on mountains, high mountains. There's certainly a lot of alpine tundra in the Himalayas, as you might guess. Coniferous forests, those are forests with needle-leaf trees. Well, not always needle-leaf, but coniferous forests. In the Himalayas, certainly a lot of coniferous forests. Temperate forests. Well, some people say broadleaf forests, but it doesn't necessarily, temperate forests can include pines and things like that, which have needle leaves, but in the temperate zone. Tropical forests, typically evergreen, although not always evergreen, in the tropical zone, where there's not enough moisture, and that's pretty much the main factor. There are grasslands. Grasslands tend to be continental, near the center of continents, as it were, and grasslands represent a very distinctive type of ecosystem, but we don't have as many grasslands as we used to. Well, not the way the natural grasslands. The grasslands of planet Earth are now the agricultural areas. Now, we still grow grasses, wheat and corn and things like that. They're grasses, but they're not the grasses that there used to be. So Myxos have been hurting a little bit with that particular ecosystem type disappearing. Deserts. Now, Myxomycetes, like any other living organism, Myxomycetes require moisture. Moisture can be very limiting in deserts, but Myxomycetes can be found under some incredibly dry conditions. They're amazing critters in that regard. Next slide. What are the major factors that influence the distribution of Myxomycetes? First, moisture. I've already mentioned that. I've referred to it a number of times. If there's no moisture present, they're going to have a tough time, simply put. The second thing, there's got to be temperatures high enough to allow them to develop and fruit. Myxomycetes do not well at very low temperatures. They also don't do well at very high temperatures, although some Myxomycetes seem to have evolved to be able to tolerate rather high temperatures in deserts. Not been studied a lot either, but we know it's true. Number 3 is a big one. Availability of suitable substrates. If something invariably is found fruiting on bark, and there's no bark around, you're not likely to find them. The best example of this is simply wood, coarse wooded debris. If you go very far north or very far south on planet Earth, you reach a point, you get beyond what we call the tree line. You go far enough north, and there are no more trees. You say, well, wait a minute, no more trees, that means there's no more wood. Start looking for Myxomycetes. The things that are associated with wood, you're not going to find, except in very exceptional cases. They're gone. They don't have the substrate. The fourth thing is something, again, that actually a little hard to quantify because we can't see spores. I know you can filter air for them. There are various ways of doing it, but getting a total picture of where spores are and where they're blown to, because we think wind disperses both spores. Well, we can't do that. But dispersal of spores, how well dispersed are spores? Now, in theory, a typical Myxomycete spore is no more than 10 microns in diameter. At that size, wind should be able to carry it all around the world. I mean, a spore of that size literally almost floats in the air. However, Myxomycetes do seem to display distribution patterns that reflect a limited distribution. Some things are found in one area of the world and have not been recorded in another area of the world. So we say that widespread or long distance dispersal, there have to be some limitations of it, but we don't know exactly what or how. Next slide. This is, the vegetation here is lush. No, it's not. This is Southern Africa, actually the country of Namibia. That desert is thought to be the oldest desert in the world. Some people think it might date back to the Cretaceous. That's kind of hard to believe, but it's certainly been dry for a very, very long time. Limited vegetation, a few plants here and there. Certainly no Myxomycetes. Wrong. We collected Myxomycetes there too. Wow. And they could survive there. That's so dry that in a given year, there's no rainfall at all, no precipitation. Pretty incredible. Next slide. Would you think this would have more Myxomycetes? I would say yes. This is a forest in northern Queensland, Australia. And absolutely no question, that looks lush. It is lush. There are lots of microhabitats. One of the few studies that has looked at Myxomycetes in canopy soil that was carried out there. It's a good place to go looking for Myxomycetes. It's a wonderful place. Australia is known for one thing that India is also known for: poisonous snakes. So you have to watch out when you do field work. Next slide. Whoa, this is a desert in South Australia. It doesn't look like it would be very inhospitable for Myxomycetes. Well, it wasn't real hospitable for Myxomycetes, but the collections I made there, well, there were three papers published on it, found enough to certainly justify talking about them. So yes, there are Myxomycetes there. Next slide. Some of you are saying, what the heck are those? Well, some of you know, those are seals. Some of you say, is he crazy? No, he's not crazy. Those are penguins. I know what they are. This is on Macquarie Island. And I had the opportunity to spend 4 months on Macquarie Island as a visiting researcher. It was an extraordinary experience, even though I was a group of people, a group of scientists, about 30 of us. We were, at the time, we were among the most isolated humans on planet Earth. But needless to say, people hadn't studied Myxomycetes there before. Well, in those 4 months, I made more than 400 collections of Myxomycetes. I mean, it's very worthwhile. Quite a place. If you like penguins, the largest penguin colony I saw, which is not this one, had an estimated 250,000 birds. 250,000 birds. Wow. I see I'm getting a few questions, but I'll ask those people to hold them until I finish, and I'll answer any question you have that I can answer. Can't swear I can answer every question. But no, Myxomycetes do not occur in penguins. I'll answer that one. Next slide. I've done a lot of work in New Zealand. It's a very pretty place, and I like it for one reason that makes it very different from India. There are no snakes. There are no snakes in all New Zealand. So you can go anywhere. You can roll on the ground, nothing to worry about. Well, in all the years I spent there, and I made 20 trips over the years, I've collected all over the place. And my collections actually give some data relating to the latitudinal distribution of Myxomycetes. In terms, you look at latitude going from the, say, the South Pole to the equator. For most organisms, we think that diversity or biodiversity or species richness, whatever term you want to use, you get more and more and more and more species as you go north, or if you're starting at North Pole, as it goes south. Well, that's rarely been documented for microorganisms, and since the Myxomycetes have well, they spend most of their life cycle as microorganisms, you classify them in that. It's really hard to get distributional information of this kind for microorganisms. But, well, I was lucky enough to get some, and you see, well, the lowest total down there, 25, and it increases more. And you get to mainland New Zealand, and it really kind of explodes all over the map. I will say something, though. Sometimes you have outliers that don't conform to that pattern. If you look very carefully on the right, you see Chatham Islands. Some of you see it, I think. Well, 6 months ago, I made a collecting trip to the Chatham Islands. I went with my trusty field assistant, a field assistant I've had for more than 50 years, my wife. And we collected the first Myxomycetes on the Chatham Islands. Chatham Islands is incredibly poor for Myxomycetes. It stays cool, it stays wet. It's just not a very good habitat for Myxomycetes. So we didn't get a lot of stuff, but I was able to collect Myxomycete data for another region of the world. And that's what research, one of the aspects of research, you collect data for a place or for a thing or a system for which data do not exist. That's the justification for scientists. Next slide. Early in a project, I was involved some years ago, it's been a couple decades ago now, in a worldwide inventory for slime molds, including mostly Myxomycetes. At that time, we tried to compile existing records to plot them on a map of the Earth to see where most of the records exist. You see those little red dots. You say, whoa, those little red dots seem to be concentrated in some places, don't they? You say, whoa, wait a minute. Look at northern North America. Where are the little red dots? Wow, look at northern Asia. Where are the little red dots? Look at half of Australia. Where are the little red dots? Look at Africa. Their whole countries. No records at all. Wow, why is that? Well, that's because people haven't looked there, simply put. Next slide. If you look at this and look really hard at the map, these are the places where Myxomycete research, good, solid Myxomycete research, very worthwhile information could be obtained. Africa, lots of places where we have very limited data. Southeast Asia, there's still white spots, blank spots in the map there. The Middle East, well, the Middle East, there are a lot of blank spots. Unfortunately, the Middle East tends to be a place where a lot of, well, I know that Americans don't necessarily want to go there because of the conflicts that are still ongoing some places. Northern Asia, well, there's certainly places there. Something that's really neat, and I wish I could do more, the small islands of the Pacific Ocean. A few years ago, well, actually it started in 1995, I became interested in island biogeography. Now, what the heck is island biogeography? Well, it's looking at islands and finding what Myxos got there, figure out how they got there and whatever else. I started off with Quarry Island. I later made visits to two other sub-Antarctic islands. I've been to Ascension Island in the middle of the Atlantic Ocean. And within the past 7 years, my wife and I have made trips to Christmas Island in the middle of in the Indian Ocean, and Norfolk Island in the Pacific Ocean, getting data on island biogeography. I've reached the point I finally have enough data to put a paper together, but the paper is not yet written, so don't expect it tomorrow or anytime soon. Northern North America, large stretches of Canada, hardly anything. Some areas of South America, not a lot there either, although there was a major project, that major project I talked about, we hit South America pretty hard. Some areas of Australia, well, since that map was compiled, I've hit Australia pretty hard. So there are a lot more red dots in Australia than there were. So this is not completely up to date, but it still gives you an impression of where we've been. I'm talking about in a collective sense, scientists, not just me, and where we still need to go. Next slide. Most of you know what fossils are. Old professors are called fossils. I'm a fossil. But there are other kinds of fossils. That's a fossil stematitis in amber that has been reliably dated to 100 million years ago, to the mid-Cretaceous. They say mid-Cretaceous, wait a minute, weren't there dinosaurs running around then? Yes, indeed there were. Wow, that's sort of the height of dinosaur evolution. Just after the Jurassic that everybody's heard about. That Myxomycete could actually be assigned to a species that is still out there today. Now, scientists don't do that. It was given a new name. I don't know, because when the paper was published, it was still unnamed. They said, wait a minute, dinosaurs, could I have gone back to the mid-Cretaceous and fill my collecting box with Myxomycetes? Yep, you could have. The only thing is, there'd been something a lot worse to look out for than snakes, right? But how far back do the Myxomycetes go, and things that are easily recognized? I mean, Myxomycetes don't fossilize easily, so we may never have any older fossils than that, but it's certainly a suggestion that they go back a long ways. Next slide. In 1994, I had a book published, Handbook of Slime Molds. That book was in print until 2 years ago. I was kind of proud of it. A lot of people used it. Next year, there will be a new version of that, rewritten, lots more pictures. I think a lot better version. A handbook of Myxomycetes, slightly different title. And if you're interested in Myxomycetes or know somebody who is, tell them about that book. Say, hey, maybe I should go and take a look at that copy, and maybe I know who wrote it and know something about him. But the publisher likes for me to do this because they publish books to make a little bit of money. Authors rarely make very much money on a book like this, but it's nice to see when I go someplace and I see this book on the shelf. Makes me feel pretty good, and I have a feeling most of you would feel the same way. Next slide. A lot of my research has been funded by the National Science Foundation of the United States. I cannot thank them enough. One of the things that you learn about research very early on is that you don't do a lot of research unless you have money or rupees or whatever else. You got to have money to go places, to buy equipment. If you don't have it, well, what can you do? I'm retired now. I have no sources of funding other than what I have of my own funding, and I'm still doing research. And I'll do research as long as I can. So I'm not saying it's an absolute limiting factor. I know some people who've done some incredible research, and they've never had a grant from NSF or a major grant from anyone else or anything else. So I encourage you to do what you can. Don't that be a limiting factor. If you have the desire, the interest of doing research, do it. And the next slide is the last slide, which is no slide. So I'm finished, and now I'm opening the floor, as it were, or the screen, or whatever else, for questions that you might have. Questions?
Thank you, Professor Steve. I hope you can hear me.
I can.
Thank you very much for that wonderful presentation. With lots of personal experiences you shared with some beautiful images of Myxomycetes. Thank you very much. We'll spend some next 15 minutes answering the questions or queries or the comments from the audience. Is that okay, Professor Steve?
I had some pretty slides of India, but I figured that most of you have seen pretty places in India, so no reason to show you them again.
Yeah, and we'll invite questions. Ladies and gentlemen, if you have any questions to Professor Steve, please do raise your hand. I hope you can hear me. Yes. Okay. Who is asking me? Okay, let me start with the discussions segment with my own question. You talked about snakes. I mean, snakes of India and snakes of Australia. So how did you deal with them when you went for sampling, Professor Steve?
You're very careful. We'll mention this. Snakes can carry the spores of Myxomycetes as they move around on the ground. Think about it. Myxomycetes all throughout litter. Snakes can pick them up. In fact, I had a student actually working with the dictyostelids, another group of Myxomycetes. She actually published a little paper on dictyostelids that were isolated from the bellies of snakes and turtles and lizards. There's a lot to this world that people don't realize.
Thank you for that. Dr. Sheetal Chaudhary, please ask your question. Dr. Sheetal, can you hear me?
Yeah.
Yeah, please do ask it.
So, good evening, everyone, and good morning to Dr. Steve. I want to ask, I have actually two questions to ask. One is, you have mentioned about there are some myxo species which which can be found in deserts. So, I was wondering how they are surviving there where there is no moisture or minimal moisture would be there. What kind of physiology they have? This is my first question. Can I go on to the second question?
Let me answer the first.
Okay, right.
Okay. You remember that life cycle that I put up way back at the beginning?
Hello? Yeah?
Myxomycetes actually have two resistant stages. I indicated that the spore, if conditions get very dry, the spore can form a microcyst and can survive under really adverse circumstances. The sporosium, in other words, if it's reached the plasmodial stage, it can survive under, again, very bad conditions. It's also true that some of the Myxomycetes that occur in deserts actually produce a pyridium, which is rather thick, which maintains moisture, which enables the spores on the inside to survive. So there are three mechanisms that are involved in allowing Myxomycetes to survive in deserts. Again, some of them seem to be adapted for being able to develop under higher temperatures than most Myxomycetes. So that's a factor too. So next question.
Yeah, so my next question is actually in continuation with this, what you said just now. That means that ecology do play a role in shaping the fruiting body, right? Am I right? Do you agree with that?
Okay, I didn't catch that question. Say again.
What?
Ask the question.
Yeah, well, I'm trying to say if myxos from desert species do shifts to some other forms of fruiting body or some other forms which can be resistant in the higher temperature conditions. That means ecology do have a role to play in shaping the fruiting body structure. Hope you get it. You get that.
Yes, there's certainly some of that. In fact, remember I talked about tropical forest and the litter layer on the forest floor being wet, which is not good for Myxomycetes. Well, the Myxomycetes occur on litter or decaying wood in the tropics tend to have long stipes to get them above the moisture, the film of moisture, a film of water. In fact, one of my colleagues and I published a paper years ago. There are some species that occur in the tropics. In fact, a lot of species occur in the tropics and also occur in the temperate zone. What we did is we measured the length of the stipe for representative specimens collected in the temperate zone and stipes collected in the tropical zone, and the stipes were significantly longer for the specimens collected in the tropics. So they were certainly responding. Now, was that an evolutionary adaptation? Almost certainly. Was it constant, or did it persist if you culture these? We didn't do that. But yes, they do respond to the different environmental conditions. In other words, the fruiting body.
Okay, Dr. Sheetal, thank you for those questions. Can you please mute yourself? We are getting some technical issue because of some raising hands continuously. Dr. Rohit Sharma, please ask your question.
Thank you, Dr. Shinoy. Thank you, Professor Stephenson. It was a very nice and extensive talk, sharing your experiences. I have a two brief questions. One is, are the Myxomycetes substrate specific?
Okay, I didn't get that question.
Are these Myxomycetes substrate specific?
Well, they're specific to climate, substrate, you name it. I mean, Myxomycetes are not generalists. They don't occur everywhere at every time. They have a definite phenology. In other words, the timing during the season. For example, in a temperate climate, you'll see things in late spring, early summer. You don't see them in the fall. A given species will develop a particular peak in terms of fruiting if you collect enough and plot out their fruiting. Substrate, in fact, when a log decays early on, if you go in a forest and you kick a log, if it's freshly fallen tree, that log is solid and you're going to hurt your toe. Go back in about 15 years and you kick the log and it'll break apart. Well, the Myxomycetes associated with coarse woody debris, in other words, a decomposition process, you'll find some species early on. In fact, there's some Myxomycetes that invariably fruit on bark, on trees, on logs that still have bark. Others will fruit when the thing is partially decomposed. When it gets to be well decomposed, there are not many Myxomycetes, but there are Myxomycetes you don't find earlier in that decomposition or that process of decomposition, the sequence of decomposition or stages thereof. So again, Myxomycetes are a lot more complicated than most people realize.
Can the one found in the log can be also found in the dunks and other things?
Again, say again, I didn't get the.
A Myxomycetes growing in a log can also grow in a cow dung or dung substrate kind of thing?
Yes. In fact, where you have compost, for example, compost piles are really good for Myxomycetes. Where you cut up timber, have sawdust, old sawdust piles are really good for Myxomycetes. So you don't really even have to have natural conditions, in other words, a natural ecosystem for them to occur.
Okay. And just a small general question, how to get the students' interest towards this group of fungi? Because the recent students, they do not have any interest in the microscopy and towards the morphology of the fungi.
Well, people who have actually taken the time to look at a series of fruiting bodies and see how beautiful they really are, how intricate. I mean, these are miniature works of art in nature. They're attracted to them. In fact, some people who end up doing research on them are attracted to them that way. I mean, can you make a Myxo a pet? Well, some people have kept a Plasmodium for a while as a pet, but I wouldn't recommend it. It's not going to last very long. But if we knew more about how important they are in natural ecosystems, that would probably generate more interest, especially on the part of microbiologists. I mean, microbiologists usually don't, I mean, these are microorganisms, but most microbiologists don't care anything about Myxomycetes. But there has been, over my lifetime, there are more people interested in Myxomycetes today than there were 50 years ago. So I think that's progress. There are actually more little books, colorful little books, usually big books just full of pictures of Myxomycetes than were available back then. I mean, there's almost nothing available back then. I mean, the one book on the Myxomycetes called the Myxomycetes about Martin and Alex Office is a thick book, highly technical book. That's the only real book I had when I started out. I mean, it's still really good. I still use it, but that wasn't much. I mean, the keys for identifying Myxomycetes were kind of marginal in places, and I had a lot of help from some people early on, and it certainly helps to have somebody who knows Myxomycetes to get you started on Myxomycetes, but that's not always practical.
Thank you, Professor Stephenson, for the treat on Myxomycetes today, and thank you, Dr. Shnoth, for the opportunity.
Thank you.
Thank you, Dr. Rohit. Professor Bhatt, would you like to ask you a question? You had one question, actually. Professor Bhatt, can you hear me?
Yeah. Thanks very much, Dr. Wonderful talk you gave, and a very inspiring one. Thanks very much. Yeah. Are there any Myxomycetes with as human allergens, spores?
Well, mostly no. I had a physician who got in touch with me some years ago. She was interested in looking at Myxomycetes as a source of allergies, in other words, people reacting to them. And if you actually take the spores of a Myxomycete and inject them into a mouse, the mouse will react. Okay. But that's unusual. That's not going to happen in nature. I hate to think how many Myxomycete spores that I've breathed in over many, many decades of working with them. If they were very deadly, I wouldn't be giving this talk.
Yeah. I
that's pretty good.
That's yeah, no, probably is true. Yeah. Correct.
Nothing to worry about. No, nothing to worry about.
Yeah, this is just a query because I feel those who are working with this fungal system, probably they become more immune to those. But it's really nice. It's a wonderful group, very pretty to look at, and Myxomycetes has very remarkable group. Yes,
I agree completely.
And your slides were astonishing, wonderfully there. Thanks very much for this talk.
Well, to everyone, I'm glad I had the opportunity to do this. I enjoy spreading the gospel, as we say here in the States, preaching about them, as it were. But yes. It doesn't hurt the group if I interest someone in Myxomycetes. I feel flattered. Actually, I'm glad to do that.
Yeah, great. Thanks very much. Thanks for your things. Thanks very much.
Thank you, Professor, for your question. Madam Mila, if I'm pronouncing your name correctly. No, we can't hear you. Can you speak loudly or fix your system?
Can you hear me now?
No, ma'am. Okay. We can't hear you. Sorry. We'll go to the next question. There were some questions in the chat box. Let me read it for. Are there any cases of Myxomycetes reported growing on lichen thallus as lichen equals microbes?
Occasionally, Myxomycetes can be collected from the thallus or lichen. There are actually a few species of Myxomycetes that produce the enzyme systems. While Myxomycetes actually can feed upon fungi, and of course, a lichen is just a lichenized fungus. So yeah, they're perfectly capable of digesting, literally, a lichen. Lichens are a lot tougher to digest than the fruiting body of, say, a mushroom, but yeah, they're perfectly capable of doing that. There's at least one Myxomycete which has been rarely collected from anything other than a lichen. So they don't seem to like lichens, but they can tolerate lichens, at least a little. But I would not go collecting from lichens if I wanted to get a large collection of Myxomycetes. I wouldn't find very many, but I would find some.
So someone asked in the chat box, what kind of Myxomycetes you find on inflorescence?
On what?
Inflorescence. Inflorescence, flower system.
You mean plants living in a house? House plants?
Inflorescence. Let me spell it for you. INFLORE.
Okay. Mostly members of the Physareales. In other words, of course, that may not mean a lot to most of you, but the Physareales, the order of Myxomycetes, have lime associated with the fruiting body. They tend to be, mostly, you know what lime is. It's that group that you find mostly on inflorescences. I wouldn't expect to find something like a stemonitis on an inflorescence, but I would expect to find a didymium or a diderma or a physarum. So there are certain Myxomycetes I would expect to find on inflorescences, but there are a lot of others that I would not. So again, selectivity, as I keep pointing out, Myxomycetes do exhibit more of a degree of specificity than most people acknowledge or realize because they study them.
Okay, Madam Beatrice Dominoni, please ask your question. Okay, no. Okay, Madam Mila has typed in her question in the chat box. So she says the talk has been very interesting. She's asking, are these Myxomycetes primarily saprobic? And she's asking, could you share a bit about their decomposing capabilities?
Well, they ingest bacteria. So, I mean, people used to consider them decomposers, saprophytes. They're not at all. So, whatever term you want to use, but they're not strictly saprophytic, no.
Thank you.
They engulf the bacteria. I mean, so it's a physical process.
Thank you. Madam Dominoni wanted to ask if you could recommend some books to delve deeper into Myxomycetes, which are already available in the market.
Well, there's one book which is, well, it's a comprehensive treatment of Myxomycetes. The second edition came out, 2 years ago. What I would suggest is type my name, Stephen, my full name is Stephen, middle initial L, Stephenson, with a PH, into Amazon and look at the books that come up, and you'll easily pick up this major monograph on Myxomycetes, biology, ecology, biogeography, the whole bit. Unfortunately, it's not an inexpensive book, but it's a best single book ever published on Myxomycetes.
Thank you for that. Last question before I let you go, Professor Steve. Do you find Myxomycetes in marine environment? That was the question from one of the participants. Do you find Myxomycetes in marine environment?
Essentially, no. There have been a couple of studies looking at mangrove forests, in other words, coastal marine areas. And you do find them, but they're actually up on the mangroves and not necessarily in the water. I do not, well, I take that back. Myxomycetes, based on isolation of DNA, not actually collecting one physically or whatever else, the sequences of didymium have been recovered from the marine environment. So presumably, didymium is, the genus didymium seems to be the most diverse and the most abundant in soils. So apparently some species maybe do get a little bit of a foothold in the marine environment, but I mean, that one report is all I'm aware of for actually in ocean water. So no, I would answer that if somebody asked me, do they occur in the marine environment? I just simply say no. Now, if the person knew a lot about Myxomycetes, I might go on and say, except for one report, but otherwise I'd say no. I mean, there's no reason to elaborate on that. So for all intents and purposes, they don't.
Thank you, Professor. Thank you, ladies and gentlemen. We are coming to the end of the session. Almost 90 minutes to the session. I'm sorry I cannot allow any more questions. Sorry about that, but you are free to send emails to Professor Stephenson, whose email address I have already shared in the chat box. Professor Stephenson will be happy to answer some of your queries. And for some of the basic questions, try Googling it. You will definitely find the answers. I would like to thank Professor Stephenson for your wonderful lecture. Thank you so much. And thank you for connecting India and India with your personal experiences. It has been a pleasure listening to you.
Well, I like India.
Thank you so much. And Professor Bhatt, if you have any final comments?
No, I don't.
No. I just wanted to say how great pleasure it is to hear such things. Yeah, thanks very much, sir. Yeah, amazingly, beautiful lecture it is. Thanks very much.
Thank you.
Yeah.
I would say the few people who I actually know who met me and know me, it was really good seeing you again. I mean, this is the next thing to being in person, I guess. But no, I certainly, Dr. Lakenpal, after seeing you again, that was a special treat.
Thank you very much. Welcome anytime to India for this excellent lecture. Thank you very much for and the ecological aspects nobody else has done. You have initiated it, you have completed them, and your work is the only work on biogeography of Myxomycetes. Heartiest congratulations.
Well, thank you again.
Thank you, Professor Lakenpal. Thank you, Professor TJ Bhatt. Thank you, Professor Sridhar Kandigere. And thank you, ladies and gentlemen who have actually participated in today's talk. You have been wonderful. Thank you for your kind support, and we look forward to your continued support and encouragement for our future events also. With this, we sign off. Professor Stephenson, enjoy your Sunday. Thank you again for your spending your Sunday with us. Thank you.
All righty.
Yeah. With this, we sign off. Thank you. Bye-bye.
Okay. Evening to you guys.
Yes. They are dinner time here. Thank you. Okay. Bye-bye. Thank you. Thank you very much, ladies and gentlemen. With this, we sign off. Bye-bye.
Thank you very much.
Thank you, Professor Lakenpal. It has been a pleasure, for interacting with you. Thank you.
Thank you.
Thank you, sir.
Thank you, everyone. Bye-bye.
I am Professor Matu Arwal.
Hello, sir. Yes.
Thank you, sir. Very nice, beautiful lectures.
Thank you so much, sir. Please continue. Please attend our future events also, sir. Thank you.